Lymphatic system is formed of lymphoid tissue which is organized into diffuse and nodular lymphoid aggregations that act to defend the body against foreign elements. The largest lymphoid aggregations are lymph nodes, spleen, tonsils, and thymus. Then lymphatic vessels. These are lymphatic capillaries, which are, in comparison to blood capillaries, larger and irregular. They are branching, thin-walled blind tubes. Their wall consists of endothelium resting on a discontinuous basal lamina. There are no pericytes in their walls, but instead small amount of collagenous connective tissue, from which anchoring filaments of collagenous fibers run perpendicular and attach to the outer surface of the endothelium in order to maintain the patency of the vessel. Large lymphatic or collecting vessels have walls formed of three coats: tunica intima, tunica media of smooth muscles, and tunica adventitia, like blood vessels, but these walls are ill-defined. Lymphatic vessels have valves, which are numerous and closer together. They are important to ensure a uniform directional flow of the lymph. Then, lymph trunks. They are two main trunks collecting lymph from lymph vessels. These are thoracic duct and right lymphatic duct. Their structure is similar to a large vein, having tunica intima lined by endothelium, resting on a basal lamina and supported by subendothelial connective tissue containing elastic fibers, which condense to form a thin internal elastic lamina. Tunica media contains predominantly circular smooth muscle fibers separated by abundant collagenous tissue. Tunica adventitia contains connective tissue which merges with the surrounding connective tissue. Function of the lymph. Lymphatic capillaries drain the interstitial fluid present between the body cells and the intercellular space. The lymph content is filtered in the lymph nodes and returns back to the venous blood via thoracic and right lymphatic ducts. Chyle, however, is a lipid containing lymph drained from the intestine. Lymph nodes are small kidney-shaped bodies present along the course of lymph vessels. They are present in groups in certain sites, as axilla, groin, and so on. Structure. Any solid organ or gland is formed of a stroma and parenchyma. The stroma of the organ is the skeleton and is formed of capsule, trabeculae, and reticular stroma, forming a network to hold the parenchyma. Capsule. It is thin except at the hilum and is formed of collagenous fibers and fibrocytes. It may contain some smooth muscle fibers. Trabeculae. They extend from the deep surface of the capsule, branch and anastomose, and provide a kind of skeleton for the lymph node. Trabeculae have the same structure as the capsule. Reticular stroma. It forms the background of the lymph node and is continuous with the capsule and trabeculae. It is made of fine reticular fibers and their associated reticular cells. They can be stained and demonstrated with silver stain. Parenchyma of lymph node consists of accumulations of diffuse and nodular lymphatic tissue enclosed in a network of reticular fibers and forms an outer cortex and inner medulla. Cortex forms a layer beneath the capsule and is subdivided by incomplete trabeculae into regular communicating compartments containing lymphatic nodules and sinuses. The cortex is usually divided into an outer cortex that lies immediately beneath the capsule and contains nodular and diffuse lymphatic tissue, and an inner or deep cortex that consists of diffuse lymphatic tissue only. There is no sharp demarcation between the two zones. P lymphocytes are present in the nodular lymphatic tissue that is in the upper part of the cortex, while T lymphocytes are found in the diffuse lymphatic tissue in the inner or the deep cortex. Lymph follicles or nodules, they are rounded structures occupying the compartments between the trabeculae. There are primary follicles formed of aggregations of small lymphocytes without a germinal center. Secondary follicles formed of aggregations of lymphocytes with a central pale germinal center formed of activated lymphocytes, mainly B cells, lymphoblasts, plasmoblasts, plasma cells, and reticular cells. The thymus-dependent zone is present in the deeper parts of the cortex, paracortex. It contains T lymphocytes migrated from the thymus. Cortical lymphocytosis. They are the spaces separating lymph follicles from the capsule and trabeculae. They are subdivided into subcapsular between the lymph follicles and the capsule, and trabecular sinuses around the trabeculae. They are lined by endothelial cells and macrophages. Medulla consists of medullary cores and medullary lymph sinuses. Medullary cores are branching lymphatic cores continuous with the cortical nodules. They have no germinal centers, and they are formed of small lymphocytes, plasmoblasts, plasma cells, and macrophages. Medullary lymph sinuses. They are the spaces between the medullary cores and the trabeculae in which lymph circulates. Plasma cells are mainly concentrated in the medullary lymph sinuses. Cells present in lymph nodes. P lymphocytes are usually concentrated in the superficial part of the cortex because they are present in the nodular lymphatic tissue. T lymphocytes are mainly present in the deep part of the cortex because they are mainly concentrated in the diffuse lymphatic tissue. Activated lymphocytes, lymphoblasts, plasmoblasts, plasma cells, macrophages, reticular cells, and endothelial cells, they are present in the lymph node and usually concentrated in the germinal center. Lymph circulation in lymph node. About 10 to 12 afferent lymphatic vessels enter the lymph node through its convex surface. Lymph circulates from the cortical to the medullary lymph sinuses to be drained by about 1 to 2 efferent lymphatic vessels, leaving at the hilum. Both afferent and efferent lymphatic vessels have valves. Those of the afferent open toward the lymph node, while those of the efferent open away from the lymph node. Blood supply of lymph node. The arteries entering the hilum branch into arterioles, pass to the cortex through the trabeculum, divide into capillaries which leave the cortex as post-capillary venules. These venules between the cortex and medulla paracortex are characterized by a special endothelium that varies from cuboidal to columnar. This may limit the loss of plasma from venules. The venules unite to form veins that live at the hilum. Function of lymph nodes. Filtration of lymph from microorganisms and foreign bodies. Formation of lymphocytes. Immunological and other P and T functions. P functions. Humoral immunity. P lymphocytes identify the specific antigens, like bacteria, for example, by the help of T lymphocytes. They become activated to large lymphocytes, then changes to plasmablast, then plasma cells, which secrete antibodies against the antigen. The antibodies reach the body tissues via the bloodstream. Other functions of B lymphocytes are: some activated B lymphocytes remain as B memory cells. T lymphocytes functions. Cell-mediated immunity or cellular immunity. Antigens activate the T lymphocytes to change to killer cells, which secrete lymphokines to destroy the antigen. There are many other functions of interacting T subgroups. Helper cells, when stimulated, interact with B lymphocytes and amplify the production of antibody and help T cytotoxic and T suppressor cells. Memory cells remain as conditioned cells. Cytotoxic or killer cells by secretion of lymphokines which destroy certain bacteria as TP, cancer cells, and causing lysis of the skin grafts and their rejection. Natural killer cells are cytotoxic and are thymic independent. T suppressor cells inhibit the functions of lymphocytes, both cellular and humoral immunity.